iGEM is all about synthetic biology. So let me start off with a brief verbal anatomy of the word synthetic biology. So synthetic means redesigning, and biology is just the study of living things with logic. So synthetic biology, the whole thing, it doesn't mean to redesign the way or the logic that we study living things, but to study how we can redesign organisms with logic and to solve problems. It's a rather new field of research than traditional bioscience. It combines with traditional bioscience concepts, so it deals with microorganisms or organisms with the quite modern techniques in engineering and concepts from engineering, and aims to redesign natural living systems at a genetic level to solve problems. So the image on the left is a engineered machine with the shape of an E. coli, which is a bacteria. So synthetic biologists use molecular tools to repackage, and to reorder genes. Genes is a short to reorder genes and insert into a bacteria, and so as to allow a new desired function to be performed within an organism. So you can see when we remove the gene, we have different outcomes on the cell. So the top one without signal, and in the middle we have orange signal, and the bottom one we have blue signal. So it's all deal with the genes to solve problems. Within an institute, there are people from all walks of life as well as different disciplines. The good thing about synthetic biology is it's not limited to synthetic biologists or biochemists. It involves engineers to build genetically engineered machines with biomedical applications. And the iGEM competition, the International Genetically Engineered Machine competition, is a student-led competition organized by the MIT in the United States. And it has been a wonderful platform bringing the community and the science together, as well as people from different disciplines. In our team, we have people from majoring in law as well. And the Giant Jamboree, we went to the Giant Jamboree in October 2016, where our team and many teams from leading universities worldwide gathered to present our innovative ideas. So this is a whole lot of people there. Can you spot us?
No.
So I guess it's over here. I don't know that either. So on the left is Canaan, and in quite in the middle is myself. So back to the iGEM. iGEM has two main aims. One is aimed at creating functional biobricks and length of DNA or genetic code that produce certain functions. The certain functions or the outcome could be a production of specific protein. So here's a one biobrick, and that's a third. This is a short sequence of DNA that codes for protein, for instance. And it could be a production of specific protein that will light up the organism when produced. This could be used as an indication of something happening around the cell, because the cells are too small to be seen by our naked eyes. Let's use cooking as an example. So biobrick is a bit like, or gene is a bit like recipes. The genetic code is a stepwise procedure that within that recipe. So the function, guess what? So the function or the outcome from this cookbook or recipe.
A dish.
Yeah, it's a is a delicious, fragrant dish to die for. Or sometimes the biobrick could code for protein that sends messages to other bacteria within the same system. So almost like a chain reaction. So you can see all the green lights there, and it activates this cell and activates another. Allowing people to mix and match genes to make microorganisms to do a whole lot of different, entirely new and different things. For example, the so-called red and green light signal, just illustrate. For example, the red or green light signal for drivers on the street, or even for running the CE election. All these in normal circumstances within the same system, different signals create different outcomes. If the protein is not present, it shows a red light and no outcomes at all, and so on. So it's like a circuit, and one activates one another. So I talked about reorder, remove, and repackaging genes, and we use and we use molecular tools, enzymes, which is also a kind of protein. For instance, you might want to prepare a meal of your very own for your date. You can then tear off a dish. So we use the blue dots on our enzymes, and we cut the genes out. So you might want to prepare a meal for your date, and you can tear off the dishes that take your fancy, tear off the recipe from the cookbook, and reorder it and glue it to give yourself a very special cookbook, three-course cookbook, and that way you will have a very special three-course meal for your date as well as a cookbook, a new and unique biobrick of your very own. So it's like almost like a different kind of biobrick within the same book. So in the past, iGEM teams had used different biobricks to create new, interesting, genetically engineered machine. In 2009, the team from Cambridge used the biobrick to make living paints from bacteria. And locally, other than the Hong Kong U team and the previous teams, in 2015, the team from CUHK, they created a soil bacterium that produces magnetosome, which can react to metal ions around the water to solve the lead in water problems in the city at that time. Other teams did other interesting things. For example, the team from Stanford in 2013, they engineered a bacterium that could survive in outer space. So there's no limitation on the project that we work on. So we are the Hong Kong iGEM team 2016, and we have a unique cookbook of our very own. In our project, we have 7 different kinds of biobricks, 5 of which results in the production of unique short DNA strands. The 5 DNA strands, so we have 5 different biobricks, 5 different genes, creating 5 different very short DNA strands. And it can be used to assemble a tetrahedron. And it is a nano device, it's very small, and it can be used for to screen for biomarkers for disease. Biomarkers could be present in a serum, blood, or other body or body fluids. We think it might be a good and the good thing about our project is we don't have to do it. The microorganism or the bacteria do that for us. So you might want to ask why we need a tetrahedron. Why not a square? Why not a rectangle? Or even just a random shape, or star perhaps? We went back to the high school mathematics syllabus. It's about geometry. So upon applied force, a tetrahedron is a shape with triangular sides. So upon an applied force, it will just topple over rather than shear. So when it comes to a square or a rectangle, it will shear and it will finally collapse. So this gives our nano device a strength that it will be stable to deal with unfavorable conditions, or we don't know whatever conditions it would be. And nevertheless, the tetrahedron is good and it has great potential to be synthesized inside the cell. So sufficient and appropriate public involvement is important to the success of projects of any kind, ranging from architectures to a tiny research project like ours. Our team thinks that synthetic biology does not exist solely in laboratories or institutes, as the discipline of science exists in the community. It is crucial for the public to know what is going on inside the laboratory and institutes, ranging from the academic issues and safety issues. The human practice, which is another element of the iGEM competition, beautifully bridges these two elements together. We did a series of activities throughout the competition, and in order to raise the awareness of the public about synthetic biology. So Canaan, which is a team member from the team, will be talking you through all these series of activities.
Okay, so human practice, these are kind of...
Oh, okay.
Human practice is less serious and less technical, so it's more for general public, okay? So for human practice, we've conducted 3 activities. The first one is the John Shield Science Exhibition, GSSE, we set up a booth there. And second one is a mentorship for a local high school, UCCKE. It's vital that you know UCCKE also joins iGEM this year. And finally, we had an interview with Professor Danny Chen. Professor Danny Chen is the Assistant Dean of the School of Biomedical Science of Hong Kong U.